Hello, my name is Marija Cvetanovic, and I'm Associate Professor at the Department of Neuroscience at University of Minnesota. My lab is interested in neurodegenerative diseases, in particular how different cell types and different brain regions contribute to the progression of disease, the onset and progression of these diseases. So the title of my talk is Cascade of Regional Dysfunction in Neurodegeneration. And in particular, in this case, I'm concerning the dysfunction between cerebellum, communication between cerebellum and prefrontal cortex. So what are neurodegenerative diseases? Neurodegenerative diseases are a group of neurological disorders, and that includes Alzheimer, Parkinson, ALS, Huntington, and spinocerebellar ataxia. And they're all characterized by a few things. One is that there is a progressive neuronal loss in different regions of the brain. So each of these diseases has a characteristic brain regions that are affected. But all of them really show symptoms of impaired cognition. They have impaired mood and movement dysfunction, despite all these different regions. And this is kind of illustrated here that in Huntington, we know the striatum is particularly impacted. In Alzheimer's disease, we again know that we have hippocampus. And in spinocerebellar ataxias, it is going to be cerebellum. However, we also know that other brain regions get affected, especially with disease progression. And that is kind of what we are addressing in this talk. So why is this important? Why do we care? Well, despite neurological disorders being extremely prevalent and getting more and more with the aging population, approximately 75% of the drugs that emerge from preclinical studies, they actually fail in phase 2 or phase 3 of clinical studies. And it's usually due to the lack of efficacy. And that kind of shows here in phase 2 and phase 3. So we, A, do not have therapies, and B, most of what we find in preclinical studies does not end up working in the clinical studies. And what I would like to propose that really the gaps in knowledge of this cellular and tissue progression of disease may contribute to the failure of clinical trials. For example, we really should know how does disease progress across different brain regions. As I said, all of these start in specific brain regions, and most of the preclinical studies are focused on that one specific region. Maybe this progression, because these brain regions exist in a constant communication with each other, maybe that communication gets impacted in diseases, and that therefore, these other brain regions, even though they look okay, their function may not be fully okay. I am particularly interested in spinocerebellar ataxia type 1. That's the disease we study in my lab. This is a dominantly inherited and fatal neurodegenerative disease, and it's caused by abnormal expansion of CAG repeats that encode for glutamine in the gene called ataxin-1. These abnormal expansions lead to the polyglutamine region, so therefore SCA1 belongs to the group of polyglutamine disorders. And another example of this disorder is Huntington disease. Patients start exhibiting symptoms usually in their 30s or 40s. That depends on the number of CAG repeats that they inherit. More CAG repeats, earlier onset. And here you can see that there are two twins, kind of to illustrate that this is inherited disease. You start having movement and balance deficits, which is another name for it is ataxia. You also have dysphagia and dysarthria, which means you have trouble swallowing and speaking. And these patients also have cognitive deficits and depression. At the pathological level, these patients have severe cerebellar atrophy and loss of Purkinje neurons, and it's illustrated in these images below of the whole cerebellum and control, and then SCA1. And you can see that there are many empty spaces in the cerebellum of SCA1 patients, which illustrate that degeneration. And then when you look a little bit more close up, where these little stars are in asterisks are indicating loss of Purkinje cells. Purkinje cells are one of the major neurons in the cerebellum. Here is all the way on the right, you have a beautiful image of Purkinje cells by Ramon and Cajal. As you can see, in patients, a lot of them are lost. Now, cerebellum, as I said, I just want to introduce it a little bit. It's mostly affected in SCA, and therefore most of the preclinical studies are looking at the cerebellum. So what is cerebellum? Cerebellum is, again, this literally means little brain. And even though it's called little brain because it's at the bottom part of your brain, it is actually containing around 80.2% of all brain neurons So it actually is, even though it's 10% of brain mass, it's way huge majority of brain neurons. And cerebellum is thought to play a role primarily in movement coordination, although there are more and more studies showing that it plays a role in reward and also cognition and mood. Another brain region that we are going to be discussing today is prefrontal cortex. Again, it's a prefrontal core part of your brain, and it is thought to be the center of high-level cognitive functions, which include decision-making, working memory, emotion, attention, and perception. And prefrontal cortex and cerebellum have a really strong bidirectional communication, and quite a lot of neurons, axons in the brain, these cortical pontine fibers, are dedicated for their communication. So why do we need that communication between the center that it has this really fast decision making and the part that coordinates movement? So perhaps best illustrated is when you watch a beautiful tennis match. So we have here tennis player Djokovic receiving the very fast serve by Nadal. So what his brain needs to be is not only just see this really fast approaching a serve, which is over 100 miles per hour, he needs also to, his brain needs to think, and he will return it in such a, the very quick decision making to return it perfectly. So it will be hard for Nadal to return it. So this is just one example of quick and fast that communication can be. Again, I study this in a disease context, and I study using mouse models of disease primarily. There are two particular mouse models we are going to be using today in the talk. One is transgenic mouse model. We call it here ataxin-1 82Q. And these mouse models, we use Purkinje cell promoter to express mutant protein. So 82 is expanded for mice, 2 is normal for mice, only in Purkinje cells. So advantage of this model, it can show us how expressing mutant protein only in Purkinje cells impact prefrontal cortex without any dysfunction in any other brain region. We're also going to use flux ataxin-1 on the bottom, 146-2Q model. This is a conditional mouse model in which we have added locks and sites surrounding the coding of the mutant ataxin-1. And this time, this is a human again ataxin-1, and it's going to be under endogenous promoters. So it's going to be expressed everywhere in the brain. So that is kind of genetically going to mimic what we see in patient. But these lock sites also allow us to delete it for specific cell types. In this case, we are going to try to delete it from Purkinje cells. So our first question is, how cerebellar dysfunction impact prefrontal cortex? So we know that these ataxin-1 mice, 82Q mice, that are expressing only Purkinje cells have cognitive alterations. We have done this before using two different tests that I will show you. One is called Barnes maze. In the Barnes maze, you put the mouse in this circular table, and then there are holes around it. And only one of it is a real hole, escape hole in which mouse can go. Mice do not like to be in the open spaces, so they like to not find that hole and hide in there. So we put mouse each day, and then they use the spatial cues in the room to try to find that hole. And then we look, how do they solve this problem? So they can solve it directly. That means they're using the spatial learning and spatial strategies. So they have developed strategy and they can recognize. And then we have corrected and long correction, which are also pretty good spatial problem-solving strategies. Or mice will have just no spatial learning, but they have this serial strategy, which means if they go around, they will eventually find the hole. And random, they really are not using any strategy. They go until they find the hole. So that shows no learning or no problem-solving capabilities. So this being able to form good strategies is indicator of prefrontal cortex function to us. And as you can see with each day, so cognitive score is given for each of these strategy, obviously direct, the best strategy, they get the highest score, and then it goes down to random, in which they get no score because there is no problem solving here. And you can see that wild-type mice with each day are increasing their strategies, so they're strategizing better and better with each day. On the other hand, SCA1 mice, the one that express only Purkinje cells, have trouble doing this, so they are not capable to respond these spatial strategies. They really maintain either completely random, which is white, or serial, which is yellow. They're also deficient in a fear recall. In this case, we put the mouse in a box, and then we habituate them, and then we start training them. So they receive the shocks. And you can see mouse respond to fear of the shock by freezing. So we measure how much they freeze. And with each shock, they freeze more. And ataxin-1 mice, the SCA1 mice that express only Purkinje cells, freeze a little bit less. And then they're less capable of recalling the fear conditioning. So after 24 hours, when we go back and test them, they freeze much less compared to the wild-type mice here shown in blue. So they have cognitive deficits. So this shows us that expressing mutant protein only in Purkinje cells in cerebellum is capable to induce cognitive deficits that might reflect prefrontal cortex function. So to look how is prefrontal cortex affected in these mice, we are looking first at cellular and synaptic alterations. This work was done by a very talented researcher in the lab, Kaelin Sbrocco, and she sliced the mouse brains and looked at the prefrontal cortex, looking first for neurons, number of neurons, to see if we have any overt neurodegenerations. She used neon antibody to stain for neuronal nuclei. And as you can see here, we haven't seen any change in neuronal density, which means that neurons in the prefrontal cortex do not degenerate in these mice. Then we look at neuronal activity using a marker, CFOS, which is kind of a marker of neuronal activity, indicating that whether neurons are more or less active if they stay in with CFOS. And indeed, we have seen a decrease in the number of active neurons or CFOS-labeled neurons in these mice. So that indicates, again, that cerebellar dysfunction can lead to decrease in the activity of prefrontal cortex neurons. We wanted to see how is this correlated with the synaptic alterations in prefrontal cortex. So we have quantified both inhibitory and excitatory puncta using staining for pre and postsynaptic markers. So VGAT and Gafferin for inhibitory, and VGLUT and PSD95 for excitatory synapses. And we have seen a significant increase in the number of inhibitory pre and postsynaptic puncta in the prefrontal cortex of these mice. It's interesting that and here is an example of image when we do for pre and postsynaptic excitatory synapses. When we look earlier in these mice at 18 weeks, we do not see again any changes in the number of the neurons and neither in the number of the synapses, but we do still see decrease in the neuronal activity in these mice, saying that there's this progression of changes in which we first have a decrease in activity of neurons that then progresses to synaptic alterations. So we also wanted to look how is prefrontal cortex changed at the molecular level. So to do that, Kaelin has dissected prefrontal cortex of these mice, and then we performed RNA sequencing. When we look at genes alterations in the prefrontal cortex of these mice, we have seen that there are around 340 differentially affected genes, and most of them were upregulated, which was very interesting because ataxin-1 in SCA1, usually most of genes are downregulated. That's the direct effect because ataxin-1 is transcriptional co-repressor. And again, here are different pathways that are affected. It's interesting that we see a pathway such as glucose transport, and we also see some changes in the stress cortisol response and also some neuroinflammatory changes in prefrontal cortex of these mice. And again, I just want to remind you that these mice are expressing mutant ataxin-1 only in the Purkinje cells in cerebellum. And from this, we concluded that chronic cerebellar dysfunction is sufficient to reduce neuronal activity, density of inhibitory synapses, and gene expression in the prefrontal cortex in these SCA1 mice. But again, ataxin-1 is expressed throughout the brain, as shown in the image below. And what we really wanted to know is how these two regions communicate in disease, meaning when both cerebellum and prefrontal cortex are expressing mutant proteins and have to deal with this mutant protein. In this first situation, prefrontal cortex didn't have to deal with the mutant protein itself. It just had to respond to the cerebellar pathology. So again, we use this other mouse model in which ataxin-1, mutant ataxin-1 is expressed throughout the brain. So expressed both in prefrontal cortex and in the cerebellum. So we first, again, look at the cell number and excitatory and inhibitory synapses. Interestingly, already at 18 weeks of age, we have seen an increase in number of neurons in these mice, which was opposite of what one may expect. And we are now pursuing to see whether this is a developmental change. We have also been able to see increase in excitatory synapses. Again, this is opposite of what we see in the other mice, and a slight decrease, but not significant decrease in inhibitory synapses at 18 weeks. When we look at the older age, later stage of disease, 28 weeks, in these mice, now you can see a significant increase in neuronal activity. And again, we don't see any more change in these excitatory synapses. However, we see a trending decrease in the inhibitory synapses, which more looks like what we have seen in the mice in which mutant ataxin-1 is expressed only in the cerebellum. At the level of genes, which have seen much more genes. So again, we dissected the prefrontal cortex of these mice, and we have found significantly more. This time we have over 1,000 genes. And surprisingly, still, most of them are upregulated, despite the fact that mutant ataxin-1 is expressed in prefrontal cortex in these mice. And we have gotten really different genes, quite a lot of synaptic and neuronal markers and neurotransmitter receptors are altered in these mice, which would indicate direct effect of ataxin-1. However, we still have neuroinflammation, and we still have stress response that may come from cerebellar dysfunction. So now, to directly test which of these genes might be caused by the effect of cerebellar dysfunction due to Purkinje cell expression of Purkinje mutant protein, we use the fact that we can delete ataxin-1 using these mice. So we have bred these mice with the mice expressing Cre recombinase in Purkinje cells, which should result in the Cre expression in Purkinje cells, and then it should cut those locks inside, leading to recombination, and there is no mutant protein in the Purkinje cells, but it's expressed everywhere else in the brain. So when we looked first at these mice, what happens with the cognitive deficits? We look at early, mid-stage, and late stage of disease because we have seen this different progression of prefrontal cortex pathology at early, mid, and late stage. So again, we have used the Barnes maze and we have used fear conditioning to compare this. So at the early stage of disease, we were really surprised. We thought that deleting mutant ataxin from Purkinje cells is going to ameliorate disease. No, it actually makes them perform worse on the Barnes maze. So these mice took longer to find that hole compared to the wild-type mice and SCA1 mice at that age. And they had trouble remembering where that hole is when we tested them. And, this continued, when we retested them 24 weeks, they were incapable of remembering where the hole was. So they had exacerbated cognitive deficits. When we again looked at the with the fear conditioning, we have again seen the worst deficits. These mice were freezing much less throughout each of the training times, and they were also having less memory when we tested them. So both in fear conditioning and in the Barnes maze, these mice have shown earlier onset and more severe cognitive deficits. Then when we look at the late disease stage, we really haven't seen much differences. So both of these mice have been deficient at 24 weeks, both in Barnes maze, but also in the fear conditioning. In the fear conditioning, we think that at this point, the SCA1 mice exhibit anxiety. They actually start freezing everywhere. So this is alternative context, which is where they shouldn't be freezing, but they still keep freezing. It seems that these mice are too chill in a way, and they are not significantly different from wild type, which would again indicate that maybe, cerebellar dysfunction is helping ameliorate anxiety at that stage. We looked at the genes that were altered in these ataxin-182Q mice. Again, just to remind you, these are the mice that express mutant protein only in Purkinje cells, and then compared to ones in which we have deleted mutant ataxin-1 from Purkinje cells. And the genes that are altered in both of these have been rescued by this deletion. So deleting ataxin-1 from Purkinje cells does ameliorate altered expression of shared genes between these two mouse models, which kind of may indicate that they are caused by Purkinje cell dysfunction. But also deleting it really alters prefrontal cortex molecular pathogenesis. So again, we have only about half of the genes are shared between SCA1 mice that express it everywhere and the SCA1 mice that express it everywhere except in the Purkinje cells. So we are looking at prefrontal cortex pathogenesis, and it significantly differs, which tells us that expression of mutant protein in the Purkinje cells has a very significant impact on the molecular pathogenesis in prefrontal cortex, which I think is pretty fascinating. All in all, what we conclude here is that we have a cascade from the second part of progressive prefrontal cortex changes in SCA1 knock-in mice. So when mutant ataxin is expressed everywhere, we have a cascade of these events. Similar kind of when it's only expressed in the Purkinje cells. But we also showed that when we delete expanded ataxin-1 from Purkinje cells in these SCA1 knock-in mice, it really impacts prefrontal cortex changes. It ameliorates some, so it makes some transcriptional changes better, but it also introduces novel changes. All in all, it tells us that expression in Purkinje cells can significantly influence how is prefrontal cortex affected in SCA1. What effect this has on the cognitive deficits? Opposite of what we expected, deleting expanded ataxin-1 from Purkinje cells exacerbated cognitive deficits early on in disease. We find this really surprising, and we find this also fascinating because it was surprising, because this may indicate that early in disease cerebellum may compensate for prefrontal cortex dysfunction to minimize behavioral changes. And then when we delete mutant ataxin-1 from these cerebellar Purkinje cells, that may alter this compensation, and therefore we may have really different changes in cerebellar communication with prefrontal cortex and therefore worse cognitive deficits. We are now exploring this in collaboration with Dr. Tim Ebner's lab at University of Minnesota. They have this cortex-wide calcium imaging, so we can look at neuronal activity while mice are behaving. And in particular, they are going through the maze and they have to make decision which way to go. So we see how different cortical regions are communicating. And then we will see how cerebellar mice and deleting from Purkinje cells or different regions is impacting this communication between different regions in order to help us understand how these regions work together in a normal, healthy mice, but also how through progression of disease is this altered to lead to the cognitive deficits, which again, we think is really important to come to the really successful and effective therapies that will target this miscommunication in disease. So I want to thank all the members of the lab, student Adam, Crystal, and Keenan, and Adri is a researcher in the lab, and two postdoctoral fellows, Dr. Emmanuel Labrada and Grangot Telduktar. These are past members of the lab, everybody in Dr. Orr lab, who are our collaborators. I want to thank Dr. Ebner's lab, especially Dr. Angela Neitz and Ruth Kauter, who are our collaborator on these new projects, all the funding agencies, and I want to help our lab catalyst, Birdie, Jumba, Gloy, and Laszlo, and I would like to thank all of you for your attention. I would be happy to answer to any of your questions over email or any way that it works. Thank you so much.